Good evening. My name is Gavin Rennie. I'm the Alumni Relations Manager at the University of St Andrews. And on behalf of the Development Department at the University, I'd like to welcome you all to tonight's Saints Talk. I'm delighted to introduce our speaker for this evening's event. Matthew Holden is a Professor of Pathogen Genomics in the School of Medicine at the University. He's also an alumnus, having graduated with a BSc in Biochemistry in 1992. Since 2017, he's been an honorary consultant at Public Health Scotland, advising on the implementation of whole genome sequencing into the Scottish microbiology reference laboratories, and also the establishment of the Scottish NHS SARS-CoV-2 sequencing service. He's an honorary consultant with Public Health Scotland, where he's the head of genomics and has been responsible for establishing the Scottish SARS-CoV-2 sequencing service and integrating genomics into the COVID-19 public health response. And since the end of the pandemic, the development of the pathogen genomics strategy for Scotland. This evening, Matt will be sharing his talk with us, DNA Detective Work, Tracing the Spread of Pathogens. And in this talk, we'll explore how whole genome sequencing, WGS, has been used to trace the spread of pathogens. Before I hand over to Professor Holden, just a bit of housekeeping from me. If you have any questions for our speaker during the event, please do pop them into the Q&A and I'll put them to our speaker at the end of his talk. I'd like to invite Professor Holden to share his talk with us now. Thank you.
Thank you very much, Gavin, and good evening to you online watching this talk. So I'm going to tell you a little bit about the work that we've been doing using whole genome sequencing as a way of tracking the spread of pathogens. So a little bit of context for this talk is basically this slide here, and indeed our recent experience. I think if I was to give this talk 4 years ago, I think probably the sort of our experience and interest in pathogens and the spread of diseases would have been a little bit more distant. But I think given the last 4 years, I think it's safe to say that we've all sort of moved into the domain of being experts in pathogens and infectious diseases, and indeed the impact that they've had. And moreover, I think also over the last few years, we've also benefited from the use of whole genome sequencing to inform our response to that. So I think as we consider the sort of the last 4 years, we've become familiar with a sort of vocabulary of describing SARS-CoV-2 as different variants, the Alpha variant, Omicron, and indeed whole genome sequencers played an instrumental role in that, and that's how we've identified and tracked the spread of the virus, and it's helped inform the response, whether that be the design of the vaccines, the vaccination program, or the sort of public health response. So I think we can all safely say that we've had a lived experience of using whole genome sequencing. But I think what we'll do with the rest of this talk is just focus a little bit more about the sort of wider use of it, and also some introductions to the sort of basics around it. So as I mentioned, that we've all sort of familiar with it, the sort of impact that a virus can have on our health and indeed our lives. But infection as a process is widespread. It's a natural part of the sort of our sort of existence. Our bodies are designed to fight off infections, but occasionally we would be susceptible. An infection is when a pathogen meets a host, whether it be the human, or it could be animals, or indeed plants or other living organisms, but we're just going to focus on humans. And the different sort of pathogens that can cause disease are essentially different microorganisms, so different types of microorganisms, whether that be bacteria or viruses such as SARS-CoV-2, or sort of parasites such as protozoa or helminth, or indeed fungi. And again, the types of pathogens are a wide variety of different types, but also the sort of types of disease and severity of diseases they cause, whether that be in this case, we have bacteria. The bacteria illustrated is Mycobacterium tuberculosis, which is responsible for TB, arguably the sort of causes the largest burden of disease of a bacterial pathogen. About 1.3 million people die a year from TB, through to other sort of serious infections caused by pathogens such as the protozoa, malaria parasites, again causes a high level of burden of disease and mortality across the globe. or sort of more hidden diseases, potentially, such as the sort of helminths, the sort of infections that are widespread in areas of the world with poor sanitation and hygiene that has a large impact on health. So the sort of idea of infection is potentially where pathogens come into contact with a host, or a vulnerable host, and they can cause disease from relatively minor to more life-threatening diseases. And it's the sort of ability to combat these that is important. And in order to do that, we often have to think about the sources of where those infections come from, whether that be potentially coming from ourselves and the microbiome, the microbes that live in and on our skin that under normal circumstances don't cause disease, but if we're vulnerable or they get to where they shouldn't be, into deeper tissues, that can cause infections. Or potentially microbes can come from the environment, from contaminated air or indeed contaminated water, again causing disease, such as sort of if we bathe in unclean water, sort of E. coli can cause infection, and other sources of pathogens that again cause infection of the food we eat. And again, there's certain pathogens that we routinely see causing outbreaks in these settings, or indeed potentially infections that come from other infected humans, such as sort of respiratory illnesses such as SARS-CoV-2, or indeed diseases that can come from animals. We describe these as sort of zoonoses, where a pathogen is associated with an animal but potentially can jump into humans and cause severe disease, and whether that be directly from animal to humans, or indeed using what we describe as vectors, often insects, that can then transmit from person to person or animal to person, such as diseases like Lyme's disease that is caused by ticks, or indeed something more severe such as bubonic plague, which is associated with the bites of fleas that carry the bacteria responsible. So in order to prevent infection, one of the principles is trying to identify and stop pathogens moving from a source into a patient, and indeed causing disease in that patient, but then also potentially stopping infection spreading from the patient to other members of the population. It's by targeting these sort of steps and preventing the spread either from the source or from the patient to other individuals that we can prevent infections spreading. And a sort of key principle of this approach is using what we would describe as epidemiological approaches, where we monitor disease. And in this case, we have a sort of a graph that portrays the incidence of disease associated with an outbreak, as we can see on the bottom from the time scale and the number of cases. And in this case, if we want to avert illness, we want to sort of intervene very early on, before the disease has had a chance to spread, or the pathogen has had a chance to spread, and identify the very early cases and target our interventions there to then avert illness later down. It's getting ahead of the curve that is crucial in preventing disease. And this is where the sort of work I'm going to describe around using genomics comes in. This is where we can potentially utilise the information that we get from a genome, and indeed through genome sequencing, to genetically fingerprint pathogens in the same way that we potentially use DNA fingerprinting to understand the relationship between sort of individuals and humans, that either we're comparing humans, or indeed in the case of forensics, linking humans to events such as crime scenes. You can use the same approaches for microorganisms to compare the relationships between microorganisms, and then potentially link them to events and identify the routes by which they spread and transmit disease. So a bit of a background introduction to genomics and whole genome sequencing and the science behind this. As a sort of discipline in science, it's a relatively new sort of area. The first genome was sequenced in 1977 by Fred Sanger, here illustrated on the left-hand side. Fred Sanger notably obtained, was awarded 2 Nobel Prizes in chemistry for his work discovering mechanisms to firstly sequence proteins, and then he also developed techniques to sequence DNA. And it was using these approaches that he and his team in Cambridge at the LMB managed to sequence the first microbe, and in this case it's a virus of bacteria, this Phi-X174, and generate the whole genome sequence for this virus. And this groundbreaking work demonstrated it was possible to take a sort of microbe, and indeed, in this case, virus, but it illustrated you could take genetic material from viruses, indeed potentially from organisms, and decode them. And this is where the science of whole genome sequencing sort of stemmed from. And if we were to just sort of describe what that whole genome sequencing is in terms of a process, as we illustrate at the bottom here, it's starting with your microorganism, or indeed it could be an organism such as a human, and it's extracting all the genetic material from the cells. It's then taking those and processing those in a laboratory to isolate the DNA and amplify it, and use a sequencing technology as the sequencing machine illustrated here, and generate digital data that then can be translated into a genetic code that describes the genome of that organism. And then from that sort of string of letters of the bases, we can take that information and then analyze it to understand and identify the genetic makeup of that organism, all the genes that are encoded in that organism, and compare that organism to other organisms to provide that DNA fingerprinting I was describing. So this is the sort of principle of genome sequencing from a point of view of what is actually a genome. As I mentioned, this is something that viruses and living organisms indeed as humans and living organisms, we have our own genomes. It essentially is the genetic material inside a cell. Our cells within our body, each copy of our cells has a genome within it. And indeed, the same is true of microbes and indeed viruses. And mostly, genome is composed of DNA, so deoxynucleic acid and ribonucleic acid. In the case of some viruses, and this is normally sort of structured and organized in chromosomes, and in the case of bacteria, they also carry what we describe as plasmids, and these come together to form a genome. And on the on the left-hand side of the image here, you can see an electron micrograph of the edge of a cell that's been burst open with chemicals in the laboratory. And what you can see from this, if you look carefully, is you can see the individual strands of DNA actually starting to come out of the cell and unravel into the sort of the extracellular medium outside the cell. And it's that sort of fine line of that you can see, the sort of strand of DNA that we use as the starting point for whole genome sequencing, and to our ability to decode it. So I mentioned that the fact that living organisms and indeed viruses have genomes. They vary considerably in size from the point of view of sort of more complex organisms such as ourselves. Our genome is approximately 3 billion base pairs in size. There are genomes of eukaryotes and indeed plants that are larger, but the size of the genome is not necessarily an indicator of the complexity. It's how that information is sort of, or that molecule is used and encodes the genetic information that's crucial. But generally, the simpler the organisms, the smaller the genome, particularly when we come down to the sort of level of microbes. If we look at the relative size of fungi and bacteria and virus, we can see that bacteria, an average bacteria, has a genome a thousandth of the size of a human genome, and indeed the SARS-CoV-2 virus is 10,000 times smaller than the human genome. And that generally suggests that there's less genetic information contained within that. But actually, the way in which that information is encoded and packaged is crucial. So even though a bacteria is 1,000 times smaller in terms of the size of the genome, they pack a lot more genes into it. In this case, your average bacteria has in this case, 10 times less genes rather than 1,000, even though the genome is 1,000 times smaller. So they pack a lot into the genome. So how do we genome sequence in the laboratory? This sort of schematic diagram just sort of guides us through that if we wanted to sequence a genome, particularly of a bacteria, our starting point would be to grow the bacteria in the laboratory in culture. Then we effectively are growing lots and lots of different cells in the laboratory. From that, we can then extract all the DNA from those cells. So we end up with a large amount of DNA. We then take that DNA that will all be joined up as these large molecules that comprise the chromosome. The first thing we need to do is break it up into smaller fragments by DNA shearing, and then we take those fragments and we prepare what we describe as a DNA library. So we process those so they can be amplified up, and then they can be modified so that then they can be loaded onto a DNA sequencing machine. And it's those small fragments, when they're loaded up onto a machine and using the sort of sequencing approaches, that we effectively replicate those fragments, and in replicating them we read the DNA, and that's how we generate the DNA within sequencing machines. It's taking those small fragments and replicating them, and as we replicate them and build the molecules up, we effectively read the DNA. And once we've undertaken that, we can gather those all those fragments. We sequence lots and lots and lots of fragments from at the same time, and we take all those fragments and reconstruct the genome to assemble and reconstruct an entire genome from all those fragments of DNA that we harvested in the laboratory. And it's that reconstructed genome which is our sort of gives us our ability to analyze and compare genomes. So we mentioned that's how we get to a genome sequence, and I've sort of illustrated a representation of a genome of a bacteria here on the left-hand side. This is actually a circular chromosome of Staphylococcus aureus, an organism which we'll revisit in a moment. This is a bacteria that lives happily in and on our skin. It composed of a genome of about 3 million base pairs, and in this case it's a single circular chromosome. So within that circular chromosome in this genome, there's 2 key bits of information that are relevant. Firstly, that each genome that we sequence is a historic record shaped by the evolutionary events that distinguish it from its ancestors, and we'll describe a little bit more of that in the next slide. And then secondly, that the genome is a genetic inventory of that organism, because it contains all the information that encodes the proteins that allow the organism to function, or indeed the bacteria to function. So by analysing the inventory, we can understand and indeed predict how it can potentially behave. So I mentioned that the genomes are historic records. And so this slide illustrates what I mean here. So if we were to take 2 bacteria and we consider these on the left-hand side, and we were to consider their origins, bacteria divide and then sort of replicate by generating ancestral cells over time. So from an ancestor, the cells will divide. At the same time, it's generating more cells that become descendants. So if we pick any 2 descendants from that sort of replication over time and compare them, we can understand the sort of and characterize the genomes in each of those isolates. Now, over time, as cells replicate, they can pick up mutations. Mutations occur typically because the cell's replication machinery doesn't function properly, or indeed their DNA is damaged by sort of chemical agents, or indeed radiation that can cause DNA damage, and that ends up creating mutations. So if we were to compare 2 descendants, even though most of their genome would be shared, over time we'll also capture those mutations. And so it's those mutations that accumulate over time that firstly allow us to, when we compare 2 cells, 2 isolates that may have come from different patients that are albeit in different locations at different times, we can trace their ancestry back to a common ancestor that they both shared. So in this case, we can look at the mutation rates and calculate the time over which the two descendants shared an ancestor. So we know from studies that have been undertaken with lots of different pathogens that microbes mutate at different rates. Some bacteria, such as Staphylococcus, pick up 4 to 5 mutations a year, whereas SARS-CoV-2 picks up mutations far more quickly. But this is important information because we can then, by comparing two different bacteria, we can then predict how long ago that they shared a common ancestor. So this is useful when we're trying to investigate when two samples that may be linked to diseases shared a common reservoir that goes back in time. So it could be a useful tool for predicting not only evolution that distinguishes two separate isolates or descendants, but it also can be useful to in forensic sort of context to look when there may be a shared exposure or a common source of an infection. And we can use that information to generate effectively what we describe as phylogenetic trees or family trees that link together the sort of mutations that we can detect that distinguish two samples that shared a common ancestry, in the same way that Darwin, when he was first considering and describing evolution, distinguished and represented the differences he saw in the finches when he was sort of describing evolutionary changes. So on the left, we can see one of Darwin's phylogenetic trees or phylogenetic representations with the sort of characteristics of the finches, where he has the sort of branches that are indicating the sort of shared functions and ancestry going back in time. And we can use the same approach when we're comparing samples that come from different patients. In the case of on the right, where we have an outbreak investigation, where the branches and the lines link back and connect samples that share mutations and indeed are genetically more closely related and share common ancestry. So the same approaches can be applied, and this is what we use to investigate outbreaks. It's taking that information that we get from the whole genome sequences to distinguish mutations, and then predicting over time how closely related they are, to then try and pin them back to shared ancestry and indeed shared exposure and reservoirs. So the other important aspect is that I mentioned that are crucial from genome sequencing is we also uncover the genes that we find within a genome. So this allows us to identify some genes that potentially are important for causing disease, such as virulence factors, and these indeed genes that may be important in providing microbes with an ability to be resistant to some of the commonly used antibiotics we use. So if we can identify the genes within within a genome, we can potentially predict how pathogens may behave clinically. So this can feed into some of the decision making about patient management and in terms of treatments within the clinic. So one group of sort of context for the spread of pathogens that I'm going to focus on is hospital-associated infections. As I mentioned, infectious diseases can spread from various sources. One particular sort of area where it's particularly problematic is within hospitals, partly because we have individuals who are vulnerable to infection, often because of either their underlying health or indeed invasive procedures, but also because we've seen in recent decades the emergence of bacteria that are particularly good at surviving and thriving in hospital settings, and concerningly also resistant to many of the commonly used antibiotics. Some of these microorganisms are one that are carried in and on our own microbiomes. So whether they're carried on people, and again, most of these, if you are carried and don't cause disease, but if they get where they shouldn't, they potentially can. Or some of the pathogens are ones that are actually adept at surviving in the environment, whether that be on surfaces, in air conditioning systems, or indeed water systems that can then be spread from those sources to patients. So the particularly challenging environment for potentially the spread of pathogens. And indeed, we see that the burden of hospital-associated infections in our hospitals, both within Scotland and indeed the UK, are particularly high. In this case, the sort of recent estimates have indicated that the cost of hospital-associated infections within Scotland alone is upwards of £40 million. If we were to sort of compare that to the impact in bed days, that's the equivalent of a single small referral hospital being solely occupied by infections caused by hospital-associated infections. So you can see it has a large burden on our healthcare system. So if we can combat these more effectively by identifying the sources and transmission in these settings, we potentially can not only reduce costs but also improve patient outcomes and well-being. So I mentioned that the hospitals are a particularly problem area for identifying infections. So classically, how do we identify the transmission of pathogens in hospitals? Typically and routinely at the moment, we rely on the sort of classic microbiology. So this is where we grow pathogens that we take from patient samples, and we identify the organism and indeed often the antimicrobial resistance sensitivity patterns. And it's this information that normally flags if we have a problem where maybe two patients are growing the same organism with the same antimicrobial resistance profiles. But this is not particularly sort of sensitive. And we've seen the sort of development of molecular typing approaches, which is starting to use DNA to identify the sort of differences between small DNA fragments. But with the advent of whole genome sequencing, we can go one step further. Rather than just relying on small bits of organisms, whether it be proteins or fragments of DNA, we can sequence the whole genome. And the benefit of this, it gives us an increased resolution to be able to compare samples that come from the same patients, and with that resolution we can distinguish very fine scale sort of portraits of those organisms to be able to compare, to identify where organisms are very, very closely related and indeed linked, that we can rule in transmission and then target our interventions accordingly, or more importantly rule out and identify where infections don't have a common source, and then repurpose and reprioritize resources accordingly. And it's this increased resolution that whole genome sequencing gives us that's particularly powerful. And so this is work that's been undertaken within St Andrews, within the group that we've undertaken here, working with partners across NHS Scotland and the reference labs, generating a capacity to sort of take clinical isolates, undertake the whole genome sequencing, and undertake the sort of analysis of the data and provide reports back to infection prevention control teams to help inform their decision making. And so to illustrate this, I'm going to just take the last part of the talk just to run through an example of a particular organism. And I think this is a good example of the power of this technology to help understand and prevent the spread of pathogens. I'm going to focus on Staphylococcus aureus. It's an organism that's particularly close to us in terms of our own microbiome. About a third of us carry this harmlessly up our noses, but it's a formidable organism because of some of the facets it has in terms of its ability to survive and thrive and cause disease. We know it has a broad host range. It's part of our natural microbiome, so it's coexisted with us over the sort of thousands of years over which we've evolved. And indeed, it can colonize other wider host range, including sort of companion animals, wild animals, and indeed sort of domestic animals. It also has an amazing ability to cause disease. As I mentioned, most of us can have it colonizing us and it doesn't cause disease, but if it gets somewhere it shouldn't, it can cause a wide range of disease, from relatively minor infections such as skin infections through to more life-threatening infections such as endocarditis and septicemia. It's also very good at generating antibiotic resistance. We've seen the emergence of antibiotic resistance in this pathogen shortly following the introduction of new antibiotics. So it's a constant challenge in terms of our ability to treat infections that are developing resistance, because the increase in the burden of resistance we see in this pathogen over time. So the numbers of antibiotics we potentially can use against some infections is diminishing because of its ability to develop resistance over time. And then finally, we've also seen the sort of emergence of sort of variants of this microorganism that are also very good at surviving and spreading within particular settings, whether that be in hospitals or in the community, or indeed in agricultural settings as well. So really, it's a fascinating microorganisms to study, but also it's a formidable problem in hospital-associated infections. So what I'm going to do is hopefully take you through an example where we use whole genome sequencing to help investigate an outbreak that occurred at the Special Care Baby Unit. In this case, the Special Care Baby Unit at Addenbrooke's down in Cambridgeshire. And we were working at the time, this was work that was done in collaboration between Addenbrooke's and the Sanger Centre down in Cambridge. And we became involved in this study because the Special Care Baby Unit identified that there were a number of babies on their ward that tested positive for Staphylococcus aureus. Indeed, this was a methicillin-resistant variant of this. And so they wanted to understand whether or not this was part of an ongoing outbreak, or this was part of just an event that had occurred recently. So in the figure here, you can see this is a schematic that shows a number of babies on the Special Care Baby Unit were screened positive for MRSA. And you can see the red circle were the 3 babies that initially instigated this investigation. But when they went back and looked through their records, they identified there were a number of other babies that had tested positive for MRSA over time. And so we were interested in investigating the relatedness of the 3 babies that had tested positive during a short space of time, and then the relatedness to all the other babies that they tested going back a number of months beforehand. So in this case, we used genome sequencing to identify or sort of characterize the genomes and compare them to identify how genetically related they were and the numbers of mutations that distinguish them. And indeed, when we investigated them, we identified that all the babies over time, and indeed some of the earlier ones that had been ruled out because they had a slightly different antibiotic resistance profile, were all genetically closely related. In this case, they were all related to each other by less than 6 mutations, as you can see by the sort of bullseye diagram, how they're illustrated by the lines that connect them back to a sort of a predicted common ancestor. So this firstly confirmed that there had been potential transmission on the ward, but actually those 3 babies that had been originally identified, this was part of an ongoing transmission that stretched back a number of months. In this case, the clinicians, the infection prevention control nurses went back and dug through their records and identified that there were a number of other cases that were identified in the hospital that had the same antibiogram. And in this case, we identified those and sequenced them, and we can see that these have now been added to the top panel at the top here. Now, some of these were other samples from, in this case, neonates and babies, and some of these were from adults as well. And again, when we genome sequence them, we can see that, again, they all started to fit into that larger family tree, albeit some of them are on longer branches. But then when we looked at the timescale, we could see that these actually were further down the line than the original investigation, indeed a number of months, and this investigation stretched over potentially a year. But nevertheless, it showed us that this was part of a transmission that linked the Special Care Baby Unit to other samples. When we looked at where those other samples came from, we could identify that some of these samples actually came from other babies that were originally on the ward but had screened originally negative but then subsequently screened positive. And indeed, some of those samples that in this case came from adults were actually from the mothers of babies that were linked to the original, whose babies were on the ward at the same time. And indeed, some of the mothers that were actually on the maternity ward at the same time. And in some cases, we had babies that were on the Special Care Baby Unit that were positive, whose mothers tested positive, And then in this case, we also identified that some of the cases were the partners of the mothers whose babies had tested positive, who then presented further down the line at GP clinics with infections associated with MRSA. So we identified that in this case, what started on the special care baby unit then spread beyond the special care baby unit into other wards in the hospitals, and indeed followed some of those cases back into the community and persisted potentially in those households over time, then resulting in disease. So when we were investigating this, it became apparent that this was actually quite an extended investigation and indeed quite complex, but whole genome sequencing was starting to provide links where, because of the timescales, we wouldn't have necessarily seen them from our sort of recording of the sort of epidemiological information. But when we were investigating this, halfway through, we also identified there had been a large gap when there had been no cases, and then we had another baby that tested positive, suggested even though there had been a gap, there was a continued spread of the infection on the ward, and indeed the isolate from that baby also clustered with this, suggesting that there was continued spread within that ward. On the basis of this, we hypothesized that there was potentially a source within the ward, and one of the potential sources was a member of staff. And so this information that we would generate from the genome sequencing was used to actually consult with members of staff to demonstrate that potentially there was a potential transmission within this. And as a result of this, and through indeed the support of the staff, there was a screening of 154 members of staff were screened, and one member of staff tested positive for this. And when we sequenced the samples from that member of staff, we can see here we've picked a number of colonies from there. In this case, we swabbed their nose and picked a number of colonies from those, and you can see all those colonies from the member of staff clustered with the baby sample, indeed linked back to the original outbreak. On the basis of this, we hypothesized that potentially they were a link in spreading the transmission, and in this case, the member of staff was put on sort of gardening leave and underwent a program of decolonization to remove the MRSA that had been effectively growing quite happily up their nose. And then once they were decolonized, the member of staff went back to work, and from that point onwards, the further surveillance of the special care baby unit identified that there were no further transmissions from this strain of MRSA on this ward, highlighting potentially that the important role here that using genome sequencing to DNA fingerprint the pathogens was crucial in helping us to understand that there was an ongoing spread, but also to identify and target interventions to stop this spreading further. So in this case, what we did was to act at this potential point where we could potentially act at this point here. In this case, because there wasn't genome sequencing, the disease spread, and we had a number of cases where what started on the special baby unit then went around the hospital and into the community. But ideally, what the work showed was if we could intervene at this earlier point, and if we use genome sequencing very early on for those first cases, we'd been able to identify there was a transmission. We could then have ultimately put in place some interventions that targeted this point here to prevent the onward spread and the sort of events that happened earlier. So this hopefully sort of illustrates very nicely that if we have a sort of an approach where we can routinely use whole genome sequencing, we can spread the transmission, we can identify transmission very early on and target our interventions accordingly. So just to sort of wrap up and put this in context, how do we use genomics to combat disease? I focused on a very sort of specific example and hospital-associated infections, but the sort of work that has been undertaken in this area have identified that whole genome sequencing potentially is important for diagnosis, so we can use it to identify outbreaks and sort of target our interventions accordingly. We can also use it for identifying new pathogens and indeed identifying the sort of reasons behind treatment failure to target our therapies more effectively. But also another key area is in population surveillance, that's sort of looking not beyond the individual patients to look at how pathogens spread in the population, and indeed the sort of emergence of high risk variants that can inform our response to those, whether that be the sort of implementation of vaccines and looking at the effectiveness of vaccines, or indeed a growing threat at the moment is the problem antimicrobial resistance. And again, just to finish off the sort of work that we've been doing in Scotland, and to bring it back to the sort of the experience of the last 4 years and where we are with genome sequencing, it is routinely used for whole genome sequencing of SARS-CoV-2 in Scotland. We use it for diagnostics, for identifying outbreaks, and also informing our use of antivirals. But also it's become a routine part of the way in which we describe the variants and monitor the variants and undertake the risk assessments that inform both the design of vaccines and also our immunisation, but also that horizon scanning of the sort of spread of the disease, and indeed potentially the emergence of new variants in the population, whether that be through sort of spread in the community and causing clusters of infection, or actually at the level of the population with wastewater surveillance. So I'm going to just end the talk by thanking some of the people who've contributed to the work over the time, in St Andrews particularly the work through shaping, and I'll stop there for questions.
That's great, Matt. Thank you very much for sharing that with us. We have had a few questions come in. If I can just read the first one out to you, please. The questions come from David, who asks, can one infer from the sequence whether a variant is likely to be resistant to antibiotics, or do you have to know the variation in the structure?
So that's a very good question. It's something that we have started to use routinely now for certain pathogens. So in Scotland, we routinely use whole genome sequencing to identify the initial antimicrobial resistance profiles of TB pathogens. That's partly because TB takes such a long time to grow and is difficult to sort of phenotype using our classic microbiology. So we now use whole genome sequencing for that. And it's effective there because over the years, we've built up such a detailed understanding of what the resistance sort of mechanisms are, what the mutations are, what the genes are. So in that case, we've got a really good catalogue that we can refer back to. But for some pathogens, it's not as effective, partly because those microorganisms are harder to predict the resistances for, but also in those cases, it's arguably quicker and cheaper just to profile them using standard sort of phenotypic methods that we use in the clinic. So for a lot of the sort of clinical testing we do, at the moment, the sort of standard approaches are just as cost effective. But in a situation where we potentially do move over whole genome sequencing, if we can generate the data quickly and we can do it cheaply, there's certainly a role for that. The one caveat to that is it tells us about the sort of potential resistances we know about, but when we have new antibiotics come along and we need to be able to acquire knowledge as to how resistance develops and the mechanisms of resistance, I think we're still going to need sort of a level of phenotypic testing, or the sort of classic testing we do in the microbiology labs to inform our knowledge of what are the sort of genetic mutations and genes we should be looking out for. So it's going to be a bit of both in the future, I think, at the moment. But certainly for certain pathogens like TB, it has been routinely used. Okay, I think it's work in progress with the other ones.
Thank you. Thank you. David actually had a follow-up question to that, which I think I'll just put to you just for 2 seconds. He also comments that Sanger has massive compute power. How can you possibly cope with that?
That's a good question. So in terms of it all comes back to the amount of data you're generating and how much data you need to get the information you want. Genome sequencing potentially is a sort of a computationally heavy process, But I think it comes down to identifying the sort of what do you need to do, what sort of information do you need, and then refining the processes and streamlining the processes. I think there's a temptation. It's easy to generate lots of data, so I think this is certainly a case for identifying what are the sort of the key bits of information you need, and whether that be what are the key sort of samples you need to sequence to address the question, or indeed the sort of methodology you want to approach to maybe on an individual level to predict the sort of sequence. So there's an element of that is making sure just don't generate data because you can, because it is computationally heavy. But the other, on the flip side of that, as the technology improves in terms of the way it generates data and the data it outputs, I think there is this move towards sort of streamlining that, just recognizing that sort of less sort of voluminous data is easy to handle, and also the sort of technology approach, the sort of compute that you now see on your average sort of mobile phone in terms of what it can do computationally is far in excess of what we would have had before to do sort of even some of the early genome sequencing. And in fact, there are sort of projects underway where you can attach sort of miniaturized sequences to mobile phones to generate sequence using the compute from a mobile phone. So wow. It is opening up new possibilities, but I think yes, it's absolutely right that it's a sort of big data project, and indeed all the data we're generating. There's some very important questions there. How do we use all that data that we're generating to actually create a sort of an informed understanding of pathogens that we can use to our benefit to help combat them in the future? And that is a big data project and big compute project.
Yeah. I find that fascinating. And actually, if I flip that for 1 second, one of the questions we've had is around our use of antibiotics. So yeah. I'm wondering, kind of, with all the data we're gathering on pathogens, could this be a tool to reduce our use of antibiotics and therefore develop a resistance to them?
Yeah, I think, well, at the first level, genomes, obviously, as I sort of mentioned, that it gives us a genomic inventory, it gives us a genetic inventory. So that allows us to undertake research that looks for potential new ways to target microbes beyond antibiotics, so novel therapeutics and novel approaches. So all the sort of information, just for example, the sort of work with things, other therapies like bacteriophage and CRISPRs, that's informed by the sort of discoveries we get from genome sequencing. So it gives us an opportunity to look for new chinks in the armor of microbes. But if we were to bring it back to the sort of the sort of antibiotic, yes, I think it does open lots of possibilities there, because potentially at an individual pathogen level, we can understand more about potentially the mechanisms by which antibiotics work, and potentially utilize other approaches, whether that be repurposing old antibiotics, using combinations of antibiotics, or indeed combinations of antibiotics and other drugs, and use the sort of genetic information we get in a sort of like a sort of a reverse approach of using the genetic material to inform that, or to actually have, and I think this is probably one of the key areas at the moment, is for antibiotic surveillance, is to understand more about how the bacterial populations are evolving over time, and which antibiotics are obviously the ones that are becoming less useful, but also at the same time potentially understanding what are the more appropriate antibiotics to inform our sort of use of antibiotics and indeed antibiotic stewardship. So it's that sort of surveillance that I think is going to be important, but doing that on a sort of global level as well, just recognizing that microbes spread very rapidly, know no borders, and can rapidly spread. So it's that combination of thinking about the antibiotics we use and using them more effectively, and the part of that will be also improving our diagnostics, and genomics will probably have a role in that, but not necessarily completely. There's lots of other novel technologies there, but I think certainly a role to play.
Okay, great, thank you so much. Thank you. Got a couple more questions. I'll just go to the first one is from Professor Andrew Wallard, who says, I'm a metrologist with some international experience of physical, chemical and biotechnological science. You must, I'm sure, trace your measurement results to the SI or other internationally accepted references. What are they, and how do you assure the international validation and traceability of your results? What ISO standards, for example, apply to your work?
That's a very good question. Indeed, that's one that from a research point of view, obviously in terms of standardization and approaches there are crucial for validation of results. It's become more of an important aspect in the sort of translational work that we now undertake. So the work, for example, we undertake within Public Health Scotland, the reference labs, we're working towards UCAS accreditation and ISO standards. Apologies, I can't remember the exact standards we're working to that goes across the board. So this encompasses obviously the laboratory steps, sort of an extension of the sort of the diagnostic accreditation that is undertaken for testing that's now also being applied for whole genome sequencing we undertake in Scotland, but also the bioinformatic work. This is just to hasten to add, this is a particularly new field for UCAS accreditation, and actually colleagues in Scotland have been sort of leading the way in this. We have UCAS accreditation for the use of genome sequencing in the E. coli reference labs, the TB reference labs, and the Salmonella reference lab, and the SARS-CoV-2 sequencing service. We're currently working towards the UCAS accreditation for not just the laboratory sides, but also the bioinformatics side. So on the other side to that, I think just to address the question's point, that we also sort of undertake sort of EQA work with international organisations to again help in the validation process. But it's a crucial element of the work going forward, especially in the clinical domain.
Brilliant. Thank you so much. Thank you. I've got another question from a former classmate of yours called Leslie Braddock, who says that she studied biochemistry with you at St Andrews and she's now a GP, for info. She says, hi, Leslie looked after a case of PVL MRSA last year, which was sequenced at Colindale. Do you think ultimately local hospital microbiology labs will be able to make use of genomics to aid HAI identification, for example, as was the case in this patient? So,
firstly, hi Leslie. It's good question, by the way. It's a very interesting question. In fact, the example I showed you, the SCABU outbreak in Cambridge, one of the notable features of that SCABU outbreak, it was a PVL positive strain, and in fact one of the sort of presentations in the cases, particularly the ones that presented in GPs, were actually skin infections. As you're probably aware, PVL, this is more clinical. PVL is often associated with skin infections. In terms of diagnostic laboratories, I think yes, this is the direction of travel. In fact, this is some of the work that we're undertaking now in Scotland with the implementation of the pathogen genomic strategies, recognising we are on a sort of journey with whole genome sequencing as I sort of charted at the beginning. It was started out as very much discovery science, and we've now moved into this domain in terms of translation. It's still a disruptive technology and it's still an expensive technology. So at the moment, the sort of developments have concentrated on sort of establishing capacity within sort of big centres, but with some of the technology I mentioned, such as the sort of the ones that we discussed about mobile phones, the sort of Oxford Nanopore, we have these sort of technologies that are portable. You can potentially sequence directly from patient samples, so it reduces the need for all those sort of complex laboratory steps. So it simplifies. You could take a sample from a patient, pop it on the sequencer, generate data. So I think we're moving into that domain. I think the next question is, how do we support that? And I think we are going to move into a situation where there will be more capacity within diagnostic labs, bringing it closer to patient. And importantly, one of the things I think that's going to be crucial there is actually it will reduce the turnaround time. So it means that clinicians like yourself and indeed teams on the ground who are using the information to maybe sort of identify outbreaks can get that information more quickly and target interventions more rapidly to get ahead of the curve. So I think we are moving that direction. I think at the moment, one of the challenges is, and particularly currently with the economic climate we are in, with the sort of budgets that are available in the NHS and more widely, it's a technology that is expensive. It's going to be disruptive. It's going to take a while to roll it out, but we're certainly sort of looking to support that in terms of the sort of infrastructure we develop in Scotland. But also, allied to that, it's the training that goes around it, is to ensure that not only can the diagnostic labs generate the data rapidly and give it to clinicians such as yourself, but actually you're also working out what is the information you need to be able to make the decisions either in the management of the patient or the outbreak, but also the sort of healthcare workers around that to understand what genome sequencing can provide and how best to use that information. So the whole body of work around there is not only sort of generating the capacity and supporting that, but also training our workforce as we go forward as this technology becomes more widely available to use it to maximum benefit for patients.
Okay. Wonderful, thank you. A couple more questions, if I think we've got a bit of time to fit them in. Peter's asked, do you think we can use this technology to forecast and predict what a pathogen is evolving into and how quickly? And can you easily spot engineered changes?
Yeah, no, that is, I think, clearly one of the areas I think that would be very powerful. I think one of the things that you're going to have to consider is the complexity of the microorganisms we're dealing with and the biology of them. I think there are certain sort of pathogens where we understand how they behave and we know what to look out for, but then there are other pathogens that are far more complex, and indeed that's why we do research on them. That said, if we take the recent experience of SARS-CoV-2, I think that is a good example, albeit it's a virus, so it's relatively simple. And just because of the intensity of research that's undertaken to tackle it, we have a very good understanding of how it causes disease and indeed what the crucial parts are. So in that case, actually, we did use genome sequencing throughout the pandemic and still do for what we would describe as horizon scanning, which is looking at the virus that's currently circulating, watching how it's changing over time, and then looking for things that we think are going to be problematic. And in fact, this goes back to our own lived experience. We'll all be familiar with the sort of announcements back in the day when a new variant was detected and there was concern about it. So this was work that was undertaken where we were gathering the genome sequencing data and effectively using that alongside the information that was coming in real time from scientific groups to try and understand how these new variants were behaving in comparison to what we'd seen before, and then providing a risk assessment. So we were doing it sort of in real time. And I think as this technology becomes more widespread for other pathogens, it will become routine. In fact, when we consider it at the moment, there's a lot of concern around avian flu. This is an area where we're working. We understand some of the risks there. We also understand some of the variants that we potentially want to be looking out for that informs our risk assessments in terms of their potential to jump from birds into mammals and indeed to humans and then become problematic. So it's that sort of, we need to build up a knowledge of understanding and also generate data that we can, genomic data that can give us the genomic intelligence, but make sure it's sort of linked back to the sort of biology, if you will, and that understanding of how it behaves.
Yeah. Okay, great, thank you. I'm gonna ask you one last question. Okay, it's from Samantha, and Samantha asks, what kind of time frame does near real time refer to? So would this be fast enough to test patients coming into wards via A&E, for example?
Yeah, no, that's a very good question. In this case, we were utilising a technology that does have a bit of a turnaround time. So in this case, we were looking at a turnaround time in about 5 days, which, when you consider the timescale and speed of outbreaks, is not in any stretch of the imagination sort of real time. That said, the technology is progressing. We mentioned some of the technologies at Oxford Nanopore. The way it works, and it's a really cool technology, it works by effectively using pores that you pull individual strands of DNA through. And as the strand of DNA is pulled through the pore, you measure the charge across the pore and you can generate data in real time. Effectively, it's pulled through the pore very rapidly. You're reading the sequence. So technologies like that, you effectively generate sequence as you sequence. And if you can process it quickly enough, you can generate data in a matter of hours. And indeed, some of those technologies you can take samples directly from a patient sample and then put them directly onto the sequencer. So in those cases, we're looking at things that, if they can be operated close to the patient, that you are getting to the point where it is almost real-time sequencing, so it could inform decision making. And indeed, there were certain cases and certain times within the pandemic where there was particularly important samples where, even with the high throughput approaches that we were generating, we were able to sequence and get data back to clinicians within 2 days of the sample coming back from a patient that could have been remotely located across Scotland. So yeah. I think that's one of the key areas to speed it up, but also to make it cheaper, because that's the other crucial thing. You can speed things up, but it's often expensive, but to make it a routine part of what we do.
Yeah, brilliant. Okay, thank you so much. I'm afraid I'm out of time to ask any more questions. Thank you so much to everyone who's submitted questions in the Q&A. We have recorded this evening's session, so that will be available to all who registered for it, and that should be going live tomorrow or certainly this week. I would just like to say thank you, Matt, for sharing your talk this evening. I've really enjoyed it and found it very enlightening, and I hope everyone else has as well. Finally, from me, I just want to say that our next Saints talk will be on the 28th of May. It will be delivered by Dr. Leshu Torchin from the Department of Film Studies. The talk will be a documentary film and other media in a post-truth world, and details on how to register will be circulated in advance. For now, I'd like to say thanks again to Professor Holden. Thank you everyone for watching, and we look forward to welcoming you on our next Saints talk. Thanks very much and good evening.